First, thank you all very much for a really terrific set of talks. And there's a lot of questions, but I'm going to try to pick the most general of these questions that I think are representative of the field in general. So a question I had for you, Rong, and maybe this is something that software issues are related to as well, is with the ability to look at all these different levels of omics, including proteins, you should be able to look at cell-cell communication networks. And I know that you can see some of this in Raman as well, but is that a technology that's coming along in tissue, look at communications?
Yeah, I think broadly speaking, probably two types of cell-cell communication. So one is directed contact. So I think in that case, if you are trying to look at proteins, we do need to kind of develop those kind of capture agents or antibodies to measure both. And I think the current panel nowadays available for kind of binder either Akoya or those kind of commercialized systems, probably still not a very good coverage. So the second type of cell-cell interaction is really kind of paracrine signaling, and that can go much longer distance. But either one, even if you are not directly measuring that cell-cell interaction, either through contact or through paracrine signaling, you can still get that information in two ways. So one, I think people are looking to either transcriptome or epigenome data, and from the transcriptome epigenome data, you can kind of find out what kind of cell-cell ligand receptor interaction genes are expressed. You can do the show up the cell ligand receptor interaction directly in the tissue. So number two, even without that information, if you just have no codex to identify cell type, you can still kind of compute something like a niches or neighborhoods. And although you don't know the exact mechanism, but got to be something underlying your observation if you constantly see that type of niches or neighborhood. I think both have been actively pursued in a field to look into cell-cell interaction. Either you know what the interaction is in terms of molecular kind of cell ligand receptor interaction, or even you don't know, you know the cells tend to be together, must be something going on to hold them together. Yeah, maybe Ruben can better answer this later.
Well, the question I had for Ruben, this came up as well, is so a lot of the sort of pristine data sets that one sees in this field are coming out of mouse. And I think largely it's because the researchers have complete control over the mice. You get to make a terrific biospecimen. You can preserve it in ideal ways. Obviously, human tissues are probably a little more relevant to oncology, but those data sets tend to be a lot less complete. A little spottier, not maybe with Rong's polyadenylation technique, they'll be more complete. But at the moment, by and large, they're less. But there are data sets out there, atlases, that are very organ specific. So from an algorithm point of view, how integrated are those, I guess, tissue atlases with the sort of computational packages you described? It seems like that would be a very helpful guide for imputation. You know, if you understanding what cell types are present when you only have incomplete information.
Yeah, I mean, I think most times people use single cell data for kind of like trying to impute missing information, right? Not necessarily the spatial data that is available. That are like two separate questions. You can try to integrate single cell data, and that's most of the time that is being used to enhance your spatial data and impute some missing genes. Integrating two spatial data sets, that's a whole other set of questions, and it's quite challenging. It's quite challenging if you do that for tissues that have a very defined, clear structure. Think about the brain, for example. That's already a challenge. Doing that for a tumor, where every tumor is so unique, I think that is currently, I haven't seen any method that even tries or approaches to do this, except if you think about more like microstructures, right? Between, for example, immune cells. And I think there is like 1 or 2 papers that try to map these like microstructures between a larger data set and a smaller data set. And then you can kind of like try to gain some insights from both technologies.
Yeah. Okay, and Lou, we had a lot of questions on dynamics. So in most of the single cell methods, you can get dynamics by pseudotime because basically your tissues are frozen. But your method is uniquely capable of looking at live systems in real time. So I wonder if you could comment a little bit on sort of the limitations of doing that kind of thing and the challenges from a technology point of view. And then I'm going to ask everybody to talk about where they think the sort of near-term future is. But I think dynamics are really relevant to this whole conversation.
Yeah, I guess that's I saw that there was a question asking about imaging-based omics versus sequence-based omics. I guess the advantage of doing imaging-based is that you do get probably higher resolution and then the dynamics. So one of the issues, of course, with the dynamics-based measurement is now you have to consider the toxicity of anything you are doing to the cells, even the laser toxicity over the long time, and also the throughput, because now you have to basically put your cells in somewhere over on the stage or in the incubator with a special microscope over time. So if you want to capture that time resolution, time information by doing a couple of samples at a time, that's I think on the omics level, the data the throughput is relatively low. So I guess how to better get more dynamic information, or one thing I think there are some people trying to do is trying to leverage also machine learning based information to try to make less point, but then try to infer the dynamics in that case. So I guess the data combined approach might also help with the throughput. And then we have to try to minimize all the laser-based damage so that we can really capture the true dynamics in terms of measuring metabolic dynamics.
So what does that mean in terms of laser-based damage? Is it if you look at cancer cells responding to a targeted inhibitor, things happen quickly, but they happen quickly on the order of like at least the current resolution, like 3 or 4 hours. And so it's not like you have to look at them continuously and photo bleach them or kill them with a laser. Is it just if you look at them once, is that enough to be toxic or what?
That's a good question. So yeah, so at 3 to 4 hour, that a short lifespan, if we don't really need to do it continuously. Also, I guess that's a question we don't know yet, is how often we have to image it in order to capture the full dynamics. But if we just do maybe 2 or 3 times over that 3 to 4 hour, I think that should be fine. But if you need to do it more often, then inevitably laser is going to induce some heat and then some of the photo damage, and then that might hurt the cells viability in a bit. And also, if it's not 3 to 4 hour time span we're talking about, but more slightly longer over a day or two, then we also have to consider how often we want to sample the data.
Right. Okay, so I'm going to come back to Ram because you walked us through a technology development that began, I don't know, your first paper you cited was probably 2015 or something like that. I think the first one I cited in my intro was 2016. Here we are 8 years later, and it's actually been breathtaking how fast this field has developed. So, we haven't seen much in the way, a little bit, but not a lot in the way of 3D methodologies. But just want to get maybe I'll ask all three of you from a computational, from metabolomic, from a multi-omics point of view, where do you think the field is headed? Where's the next bottleneck breakthrough?
Yeah, thank you, Jim. Maybe I can kind of offer a little bit of my own humble opinion about the 3D. I really think the imaging technologies have their distinct advantage to do 3D, and I think a light sheet microscopy and those kind of tissue clearance, when combined, you do get a pretty large volume 3D imaging of anatomic structure and some selected proteins or genes. But the distinct trade-off is the coverage across the genome, across the whole transcriptome, right? So this kind of distinct trade-off, it depends on what you want to get, right? So you want, if you want a kind of genome-wide data for mechanistic discovery, or you want to kind of resolve the fine structure that might kind of underline the specific kind of subsidiary or even single organelle mechanism of action. So it depends on the question you want to look at. But also want to kind of shout out very exciting work from one of my collaborators, Danny Schwartz. I remember like a couple of years ago, NCI released RFA to do 3D tumor atlas, and they were specifically kind of asking, okay, if you get a serial tissue sections, stack them together, that's not eligible for that call, but it turns out that's a very good idea. And then is basically kind of scan hundreds of H&E tissue sections and use machine learning AI to register them together to create extremely large scale, like a centimeter scale 3D tumor cell atlas. You miss some data points there, but that gives a pretty good large volume holistic view of the tumor. And so I would say there are many kind of ongoing efforts out there to take your tumor 3D, but each has pros and cons. It depends on the question you want to ask.
It's actually amazing to even think about it,
Yeah.
especially from where we were a few years ago. So Ruben, maybe you comment on where you think the bottleneck challenges are for software. And I know a lot of people in the software community talk about democratizing software, making it really easy for experimentalists to use, maybe easy for clinicians to use. I don't think we're quite there yet, but I'll let you talk about whatever future bottleneck you think is important.
Yeah, I would like to say the 3D is also something that we do in our lab, and I think it's super important, especially looking at, like, for example, patient biopsies. You really understand the complexity only when you look at the 3D. In terms of like software bottlenecks that I see, thinking about, again, what we're doing in the lab, what others are doing in the lab. Nowadays, we're looking at, usually you show like 1 or 2 data sets, sometimes 4 or 5. Now people are making like data sets that with 10 different samples, 20 different samples. They're using these imaging-based technologies with like terabytes of data. How do you analyze that? You can go to a server, which is great if you have that infrastructure available. Not everybody has that. So how do you democratize that process? We are working onto something that I didn't show today, which is, I think, something that you can do scalable data analysis in the future on your own local machine using like more a database backend. Hopefully that can solve some of that issue in terms of scalability. But that's definitely a huge bottleneck. Like, how can you do this type of analysis in a scalable way? And it's still in a very interactive way. And then going back to your last point, how do you make that available to clinicians, to pathologists that we are working with? Nowadays is a very clumsy process where we ask them to kind of like highlight some regions in the tissues that we work with. But ideally, you also need to work on like some actual software digital platforms, right, where they can easily go with an app and kind of like highlight what we do and kind of go back and forth, maybe in a cloud or something where you can all share and analyze the same data. But that's more like future work.
Yeah. Okay, and finally, I'll come back to Lou. So, Raman and metabolomic imaging is in some ways a step cousin of the rest of the field because you're not using a sequencing tool to get your data. And so you have to find other ways to integrate the types of imaging that you're collecting with, you know, other known biology, which is almost always coming off of some sequencing machine these days. So, and I know we did that a little bit in the melanoma paper you talked about, but how do you see that evolving over time, that the integration of metabolic imaging with these other types of omics?
That's a great question. I think one way we can do, I guess, is we also talk about it. The sort of advantage of doing RAMA is the sample prep is really minimum, right? So we can always do a sequential based RAMA imaging first, and then use a sample to do sequence based, and then we can sort of align the imaging together to correlate information. So in this case, probably we can correlate the spatial relevant information together. So in addition to what we are doing now is we get the spatial RAMA, and then we do bulk level transcriptomics or single cell transcriptomics, and then we try to mine the information through data analysis. I guess those two approach are all possible, but that really needs some experimental validation on that. So, well, hopefully we can work together to push that too.
Yeah, I mean, if you look in, like, let's say, take a single cell analysis of a tumor tissue or even of immune cells, PBMCs, every major immune cell class, as well as immune cell phenotypes, has their own distinct metabolomic character in ways that, you know, you think IL-2 is associated with T cells, but no one has a clue as to what metabolites are associated with T cells. But in fact, there are. And there's metabolites associated and all this stuff. And so it's basically a set of glasses that we just don't have a view on yet, that I think is actually pretty important for understanding tissue function.
Yeah.
Okay, anyway, I think we need to pause this session, and we have a little break before we pick up the next one. So thank you all very, very much for your talks. That was terrific. The talks will be available and archived online. For those of you who are attending this symposium and want to have access to those. And of course, Rongs and his group will be back at the end of this symposium, end of the workshop, to give us tutorials and some of the tool sets that we've heard about. So thank you all.